Hi guys, welcome to the class. So today we are going to learn about yet another interesting topic, transgenic animals. So the topic that will be covered today is transgenic animal. So what do you mean by the term transgenic animals? As the name suggests, I am transferring a gene into an animal which normally it was not having it. Like for example, this is one animal. Imagine one unicellular animal. It is having its own DNA, plasmid and all will be present if it is a bacteria. So into it, what I am going to do is that I am going to insert a new gene into it. So in case if I am introducing a new gene into it, so it will be coding for that gene which is not usually present in that animal. So that becomes what? A transgenic animal. So I am transferring a gene into an animal and making it means making it develop the protein what it is going to synthesize because of that gene, and I am developing a new animal which is having some characteristics other than the normal ones. So that one we call it as what? We call them as transgenic animals. So hope it is clear what is transgenic animals. Animals that have an extra DNA manipulated to possess an extra foreign gene called as what? Transgenic animals, right? So one animal is normal. Into it if I introduce any foreign DNA, then it becomes what? Our DNA, as we have already learnt. Introducing into it, then that becomes transgenic animals. So transgenic plants we have mostly covered. Now we are going to see transgenic animals. So it is then another application of what? Biotechnology. Now first characteristics means first step when you are saying for developing transgenic animals, for some reasons we are making these animals, isn't it? So the first step is it is for normal physiology and development, for studying the normal physiology and development. I will make it more clear. So what is normal physiology and development? What does that mean? Now I want to learn about a gene which is coding for some factor. Like for example, whether it can be an insulin or it can be lipase enzyme. So this gene, how is it functioning? How this lipase is produced? This gene, how much they are producing? How it is produced? If it is produced, what are the chemical? How the role, the whole synthesis, metabolic pathway? I can learn by introducing this gene into a organism. Like for example, I want to learn the characteristics of lipase enzyme, because this lipase enzyme I want to introduce it into a bacteria, and I want to make that bacteria produce this lipase enzyme so that I can extract this lipase out of it and can use it for making detergents. And as you know, they are good for solubilizing what? That fats or oil can be get rid of with the help of this lipase, because they digest the fats, isn't it? So like that I want to learn the growth hormone, how it is being regulated, which gene is regulating it, or any gene, how it is functioning, how the gene is functioning, that I want to learn. So in order to learn, I can make what? Transgenic animal. So what I am going to do is that I am going to take that particular gene, DNA sequence. I am going to take out that DNA sequence. For example, this is a DNA sequence which is controlling growth. What it is controlling? It is controlling the growth of an organism. Now I want to learn how this gene is controlling the growth of an organism. So what I will be doing? So immediately I cannot learn this one in putting into a human being, isn't it? So that is not done. So usually we use mice. What we are going to develop? We are going to develop transgenic mice. So this mice, when you are saying the rat, so into that I am going to introduce this gene. Into its DNA I am going to introduce this sort of DNA into this gene, means into the DNA of the rat. Now what will happen? This introduced DNA will be producing what? This growth regulating factor. Now this growth regulating factor will be produced. I will be learning the pathway, how it is produced, how much it is produced, what is its role, whether the mice is growing properly or not. Everything out of that gene I can learn where? I can learn with the help of this mice. So anything I have to develop, I have to test it first, isn't it? So for that we are making what? Transgenic organism. By introducing that gene, I am learning about that gene. Okay. So that one we call it as what? Transgenic animals, studying normal physiology and development. What is the function of this particular gene and how the thing is being carried out, what is the metabolism involved, what is the chemical steps involved, everything can be learned with the help of what? This transgenic mice, right? Now, so I will read out normal physiology and development. Transgenic animals can be specifically we are designing these animals to allow the study of how genes are regulated, how the gene is, what is the gene sequence, what is the mRNA produced, the protein produced, how much it is produced, what is the metabolic pathway, all these things we can learn, okay, and how they affect the normal function of the body and its development. If this gene is present, the mice is growing in how much, if this gene is absent, the normal one, how the growth is affected. So like that we can learn, okay, whether it is applicable or not, whether it is working or not, whether actually the mice is growing or not because of this particular gene. All these things we can learn with the help of what? This transgenic animals, okay. So that is number one. Second is your study of diseases. So what is the second step? We are going to learn about the diseases. So some of the diseases when you are seeing, they are because of some genetic defects, right? Other some diseases may be because of some pathogen is getting entry into our body and we are developing the diseases. So what we can do is that we can develop transgenic animals for the specifically when it comes with the genetic defect. What we can do, we can make the transgenic animals to learn about the particular type of disease, okay. So this is the genetic defect which is causing, maybe this is the gene, like for example if I am saying this is the gene which is causing the disease, okay. So imagine this gene is having some mutation in it and because of this that patient or the one is suffering from the disease. So I can develop the mice to study the diseases, this gene, what it is producing, okay, whether it is producing something or not, because of which that disease is being noted. So like that I can learn about the disease with the help of what? This transgenic animals. By introducing the disease causing gene into it, I can learn about the genetic defect, the pathway which is involved in that, the product which we are obtained, which they are producing, which is harmful for us. We can learn with the help of what? This transgenic organisms, okay. So many transgenic animals are designed to increase our understanding. So we are understanding better. Why? Because we are learning, we are introducing that gene into an animal and I am watching that animal, what is the effect in them, how they are suffering, what are the symptoms which are produced, okay. All these things I can learn from observing the animal in close under the laboratory condition, okay. So understanding of how gene contribute to the development of the disease, okay. So how the gene is responsible for causing that disease, what is the product obtained and what are the symptoms which are developed because of the same. Like that I can learn about the disease by using what? This transgenic animals, right? And the next thing is that we are going to develop biological products. So first is I to understand how the gene is functioning, okay, normal physiology and development. Physiology means what? Functioning, how it is developing. That was the first point, isn't it? What was the second point? In order to study the nature of the gene which is causing diseases, so that I can know more about the diseases, okay. So I can get a clear picture about the diseases, the symptoms which is caused. Based on that I can go for some treatment methods, okay, that we will be discussing. Alright, now moving on to the next point is that biological products. What do you mean by biological products? I am getting some useful substances with the help of this transgenic organisms. Like for example, a good example is your Rosie, a cow, okay. The first transgenic cow was developed in the year what? 1997. So this Rosie, what it was having is that Rosie, that cow was having a gene which is responsible for production of human enriched, protein enriched milk, okay. So infants means those who are not getting proper milk from the mother, maybe because of some various reasons mother may not be have that healthy enough, or mother would have passed away. In that case also, for the infants, the mother's milk is very, very important, because because that is making them giving immunity for the same, isn't it? So they should be given this mother's milk. So this milk, when you are seeing, cow's milk cannot compensate equivalent to the mother's milk, right, human milk. So in that case, what they did is that they developed a cow, transgenic cow. Okay, the name of the cow was what? Rosie. And this Rosie was producing human protein, which is needed for the development growth of the human. Okay, how much it was producing? It was producing 2.4 grams per liter. In 1 liter, how much was there? That protein 2.4 gram was there. So that milk is containing human alpha lactalbumin. Okay, and this is a very nutritional supplement, protein supplement, which is given to the baby in case if this milk is taken. So like that, what we developed is that we developed transgenic cow. What we developed? Transgenic cow. So in this cow, what was introduced? The gene which is coding for human alpha lactalbumin. Okay, what is the characteristics of this particular DNA sequence? This DNA sequence, or this gene, is producing a protein which is acting as a nutrient supplement for the baby. Okay, so like that, this what was developed? Transgenic cow was developed. Like that, you can find many other introduction was also done with the help of this rDNA technology. We will learn one by one. Transgenic animals that produce useful biological products can be created by introduction of portion of a DNA which codes for a particular product, such as human protein. I told you human protein. So if I am making an organism, okay, a cow or whatever organisms, cow means okay, milk I am drinking, so that part will be there, isn't it? So the first one, when you are saying, it was alpha antitrypsin. What is it? Alpha 1 antitrypsin, and this is introduced into an organism. Like for example, if you take it as a cow, it is introduced there. For what? For what? Or into a microorganism this is introduced, so that they are producing what? They are producing this human protein alpha 1 antitrypsin. And what is the advantage of this alpha 1 antitrypsin is that if this gene is introduced into a microorganism and is being cultured, this biological product, what I am getting, that is alpha and 1 antitrypsin, can be used for treating emphysema. Emphysema, you know, it is a respiratory disorder. So for treating emphysema, we can use this alpha 1 antitrypsin. So we are making transgenic organisms which are having the gene into it, which I am putting the gene which is coding for what? Alpha 1 antitrypsin, and this alpha 1 antitrypsin is extracted. Okay, and that is used for treating what? Emphysema. Okay, like that, there are many genes which are having some genetic defects. So these genetic defects are caused because that particular protein is not formed, because of which they are suffering from the disease. Like for example, phenylketonuria, when you are saying it is produced in excess, is getting stored up somewhere. Cystic fibrosis, all these are genetic defects. It can be cured by introducing a particular gene into it which are coding for those defective characters and making it proper. Okay, so for these, treating all these things, we can make what? This transgenic organisms, make that protein, and we can use it. Phenylketonuria, what is the disease? When you are seeing that, in that disease particularly, when you see the growth will be affected mentally, the person will be the patient, mostly in infants it will develop. Okay, the growth will be very less, mentally somewhat retarded in nature. Okay, and they will be having some dusty smell, in the sweat, the mucus what they are secreting, even in the urine and all, there will be some particular odor. Okay, so that is regarding phenylketonuria. And another one is your cystic fibrosis, especially when you are seeing here the respiratory system and the digestive system is getting affected. Okay, so these are the diseases which can be treated by making transgenic organisms. So these transgenic organisms are producing the product. That product can be used for treating the diseases. Okay, so for making such biological products, different biological products for treating diseases, for supplement, as food supplement, for making food supplement, we are using these transgenic organism. The best example what usually comes is your rosy, okay, which is used, which is rich in what? This rosy is rich in human alpha lactalbumin, okay. So this is a protein which is good for the growth of the baby, infant, okay. And the rosy cow, when you are saying it is having this gene into it, we are introducing with the help of rDNA technology, and this transgenic cow is producing the milk which is rich in this protein and can supplement mother's milk to some extent, not fully but to some extent it can supplement, okay. So that is regarding your biological products. So that is done. So we till now what we have discussed, it is used for studying normal physiology and development, okay. So हम लोग ट्रांसजेनिक एनिमल बना रहे हैं ताकि हम उसका नॉर्मल फिजियोलॉजी कैसे वो जीन काम कर रहे हैं हम देख सके. स्टडी ऑफ डिजीज़, डिजीज़ को इस एक जीन के वजह से कुछ डिजीज़ होता है तो उस जीन को इंट्रोड्यूस करके एक तो एनिमल में इंट्रोड्यूस करके राइज मीन्स इन माइज और रैट्स में इंट्रोड्यूस करके हम देख रहे हैं कैसे वो डिजीज़ कॉज कर रहे हैं. थर्ड, फॉर मेकिंग, कुछ चीज़ बनाने के लिए, बायोलॉजिकल प्रोडक्ट्स बनाने के लिए जो जीन उस बायोलॉजिकल प्रोडक्ट को बना रहे हैं उस जीन को हम एनिमल्स में इंट्रोड्यूस करके उसका प्रोडक्ट को निकाल के हम यूज़ कर रहे हैं, ओके. सो लाइक दैट व्हेन यू आर सीइंग वी आर यूजिंग द डिफरेंट वेज, ओके. Now moving on to the next is your vaccine safety. 5 points are there, 2 more to go, okay. So vaccine safety. So what do you mean by vaccine safety? Vaccine safety is that like for example, you know what is a vaccine, isn't it? It is a weakened or killed germs are introduced into your body so that you are getting immunity against a disease. Vaccine we have already learnt, isn't it, in human health and diseases. So so vaccines, when you are seeing, just like that we cannot introduce the vaccine into the human population all of a sudden, isn't it? We have to first test it. So for testing that one we are making what? Transgenic mice. So transgenic mice are being made, okay, coding with some DNA sequences, rDNA sequences equivalent to that of the humans are being made, and this vaccine is introduced into the mice, okay. After introducing into the mice, we are checking whether this vaccine is properly functioning, okay, in the mice or not, whether it is causing some dangerous effect, whether the mice can survive, whether it is a pros or a con, whether it is useful or harmful, we are learning. Thereafter only it is used with what? Human population, okay. So we are developing transgenic organisms, mice, okay, which is used for what? Testing the safety of vaccines. So vaccines are introduced into their body first, tested whether it is working properly or not, then only it is given to what? Humans, okay. So that is the next point in which transgenic animals are playing a major role. Next is your chemical safety testing. So that is the last point. Now chemical safety testing. So in that what they are doing? This is known as toxicity or safety testing. Like we are going to test drugs, how it is going to affect us, okay. So drugs, just like that we cannot introduce into a patient. So drugs has to be first tested, isn't it? So for that testing also we are making transgenic animals for testing the effect of drugs, whether the drugs is good, whether the drugs is compatible, whether we can give that drugs to the patient or not, okay. First we have to test it. So how we have to test it? We cannot test it in a person or test it in an organism which is immune. Immune means what? They are having good immune system. Whatever you put into their body, they can adapt to it, they will not be getting attacked, that is immune, okay. Means they are resistant, they are carrying features which are resistant. Whatever substances goes into their body, it is not going to cause a harm all of a sudden, okay. So then we call them as what? The resistant varieties. Other one is what? Sensitive varieties. Sensitive means what? Whatever the substances get into their body, what will happen? They will be causing some diseases, symptoms will be coming and they will be suffering from the disease. So those organisms are sensitive. Sensitive means what? They get attacked by substances of it, or whether it is a disease, whether it is a chemical, whatever enters into their body, they cannot adapt to it. They are very sensitive people, they get attacked soon, okay. So I am going to develop transgenic organisms which are carrying sensitive genes. What they are going to carry? They are carrying sensitive genes. So sensitive genes means what? If that organism, the drugs or the toxic substances is exposed, then that organism will respond to it immediately, okay. And what is the response? Whether it is good or not, whether it is okay or not, that we have to test it first, isn't it? So it is causing a dangerous effect or not. Okay, if the same thing, if I am doing it in a resistant variety, what will happen? The drugs will be given, he will be okay, in the meantime there will not be any problem, so we will think that the drug is okay. Then we will go and give it to the human population. The population, most of the people will be sensitive to that drug. It may not be resistant, so what will happen? That will cause a side effect in them, isn't it? So in order to test that one, what we are going to do is that we are making transgenic animals which are carrying genes which are sensitive to toxic substances. Okay. After that, in that we are going to expose what? These toxic substances. So this is the sensitive variety of transgenic organism, and this is the normal variety. Okay, normal means what? The immune variety. I have not introduced any gene into it. So here the sensitive gene which is sensitive to that toxin is introduced. Now this toxin is given to this one, as well as this toxin is given to this one. Okay, exposed. Which one will start reacting immediately? This sensitive gene will be starting to react immediately to the toxic substances, and they will give some symptoms. This symptoms is studied, whether the symptom is affordable, whether the symptoms is okay, whether or not they are not causing any dangerous effects. All these things are studied. After that only we will check whether it's okay. Mild symptoms, it's fine, but it should not create what? A very great side effect which can affect the whole population, isn't it? So that is studied. After studying this only, if the sensitive one is also responding to that toxin or the drug in a positive way, not causing much problem, means that drug is safe to be used and we can introduce it as a drug in the market for sale. Okay. So for that chemical safety testing also, what we are using? We are using this transgenic animals. हिंदी में भी बोल रहे हैं. सो ट्रांसजेनिक एनिमल्स केमिकल सेफ्टी में कैसे यूज़ कर रहे हैं? हम दो मीन्स व्हेन यू आर सीइंग, एक तो ट्रांसजेनिक एनिमल हम बना रहे हैं जिसमें सेंसिटिव जीन हो. सेंसिटिव का मतलब क्या है? दे आर गेटिंग अटैक्ड बाय द डिजीज़. ओके, सो द टॉक्सिक सब्सटेंसेस उसमें डालोगे, जैसे कि हम सेंसिटिव है तो हमारे भीतर कुछ भी आए हम उसको रिस्पॉन्स जल्दी से करेंगे. तुम सोचो कि इम्यून हो, तुम रेस्पॉन्ड नहीं करोगे. ठीक है. So what will happen is that when that germ is introduced into my body, or the toxic substances is given to this sensitive guy, that sensitive guy will react. उस reaction को हम पढ़ेंगे. उस reaction को, इस drug को जब हम इस animal में दे रहे हैं, sensitive animal में दे रहे हैं, जब हम पढ़ेंगे वो danger effects नहीं कर रहा है, मतलब वो drug ठीक है, हम यूज़ कर सकते हैं as a drug for treating diseases. Okay. And the other one we should not use, the resistant varieties. क्योंकि क्या होगा? resistant variety में यदि चलेंगे वो तो ठीक हो जाएगा, लेकिन एक साइड ड्रग जो मीन्स सेंसिटिव लोगों पे देंगे तो क्या हो जाएगा? उसका साइड इफेक्ट्स भी ज़्यादा हो सकता है. So in order to test the drugs, we are making transgenic animals which are sensitive to that drug, and then testing whether they are not having any dangerous effects, then we are using the drug for the human population. Okay. So these are the various ways which by which transgenic animals can be used in the field of medicines as well as for learning different things. Okay. So first is normal physiology and development. उसके नॉर्मल काम कैसे कर रहे हैं, कैसे डेवलप हो रहा है, उस जीन का क्या काम है पढ़ने के लिए, डिजीज़ेस को पढ़ने के लिए, बायोलॉजिकल प्रोडक्ट्स बनाने के लिए, वैक्सीन को टेस्ट करने के लिए हम बना रहे हैं. वैक्सीन ठीक है, अच्छा है, यूज़ कर सकते हैं कि नहीं, उसके लिए. और लास्ट वाला क्या है? फॉर मेकिंग, फॉर टेस्टिंग द ड्रग्स और केमिकल्स वेदर इट इज़ गुड और नॉट. फॉर दैट ऑल्सो वी आर यूजिंग व्हाट? दिस ट्रांसजेनिक एनिमल्स. So only one part of the chapter is left, that is ethical issues, that we will be covering in the next class. Hope you have understood today's concept. Thank you.